this is my presentation, and it is about the process of DNA extraction. My name is Eva, if we haven't met, and yeah, that's basically it. So DNA extraction is something that I do at my job in the hospital quite a lot, which is why I chose this topic. And I also could see myself teaching it in the future because it encapsulates a lot of very relevant things about biochemistry that I think I would aim to teach in my science classroom, while also being like a fairly cheap and easy and fun tactile activity that produces pretty tangible results. So yeah. So this is probably a refresher course for you guys, but I tried to design this as though I were teaching it to children. So DNA, or deoxyribose nucleic acid, is the hereditary information of all living organisms. It's passed from parent to offspring, allowing all of the processes of life to repeat and evolve over millions of years with consistency. Without it, our bodies wouldn't know how to function. It's like an instruction manual, essentially, that dictates everything from how to grow organs, to how to digest food, to how to breathe air. Anything that our body does, DNA is in control of doing. It is because of DNA that human beings and also the rest of the living clades of life are able to experience the similarities and differences that we do. Very much of the genetic code is the same between human beings, but each of us has a slightly different genetic sequence, which gives rise to all of the small and also large differences we see, like hair color, for example. DNA is composed of long chains of nucleotide blocks, which are each composed of one sugar molecule, one phosphate group, and one of 4 coding groups called nitrogenous bases, because all of them contain the element nitrogen, which pair together to form a twisted ladder shape that we call the double helix. These blocks have potentially been rearranged in over 108 billion plus slightly different ways over the course of human history. And the nucleotide blocks, the 4 are called adenine, thymine, guanine, and cytosine. DNA is located in every cell of our body. There is a full copy of all of our DNA in each cell. In total, there's about 3 billion total base pair blocks in one complete set of human DNA. There are approximately 10 billion total cells in the human body, so it adds up to a lot of DNA very quickly. But luckily, our cells have regulation mechanisms that prevent all of our DNA from being expressed in every cell, or we would pretty much be completely unable to live or function. If we were able to somehow extract all of our DNA at once and line it up end to end, it would be approximately the length of the perimeter of the entire solar system twice. So there's quite a lot. Each human cell is enclosed by a layer of fat to keep the watery inside separate from the watery outsides. In order to extract DNA, we first have to break apart this fat layer and then separate the pure DNA from the other things inside of cells. Luckily, we can do that easily with common household objects. So, the materials that you would need to extract DNA at home if you wanted to, or in a classroom, as I am pretending I am, is sample saliva, Gatorade, which is useful because it has a high salt content, cold isopropyl alcohol, detergent, small cups, glass or wood skewers, although glass are really preferred, and then glass tubes to perform the actual extraction in. The reason that we need the detergent is to break up that fatty bilayer, as I mentioned. That's very crucial. So basically the way that extraction proceeds is you would have the student vigorously swish Gatorade in their mouths, like mouthwash, for at least 2 minutes. That serves the function of just breaking up first of all, breaking cells off of the tissue layer of our cheeks, and then also breaking the cells up a little bit so that we are more able to remove that fatty bilayer. And then you would spit the Gatorade into the small tube, enough to make it approximately 1/3 full, although this is all, you can eyeball this, it doesn't have to be precise. And then you fill the tube to about the halfway point with dish soap, shake the mixture gently, avoid making bubbles as much as possible, and then lightly pour ice cold alcohol into the solution to make a layer on top, and then let it stand for about a minute. It's very important that you lightly pour this alcohol, and when I say that, I mean literally drizzle it on the side of the tube so it very, very gently trickles down into the rest of the mixture, because you don't want to break the DNA molecule up, seeing as it's very delicate in its extracted state. If all goes well, you should see a white layer of goo between the soap and the Gatorade and the alcohol, and that's your own DNA. You can gently pick it up with a glass skewer. As I said, it's a little bit delicate, but if you pull it out into the air, it'll immediately start to coagulate, and it looks a lot like mucus, which is interesting. As I was saying, this is what extracted DNA looks like in solution. As you can see, it's that really clear, kind of gooey looking block in between the layer of alcohol, which floats on top, and the layer of soap and Gatorade or salt water, whatever you want to use, that floats on the bottom. They separate because of a density differential, which is why it all kind of isolates in one spot. And then, as I said, you can kind of see it in the most leftward picture, but if you actually pull that out of solution, it starts to sort of condense and coagulate a lot more and looks more like mucus and less like a cloud of lace. So it's just a little interesting difference. So yeah, this works, as I said, because the soap and the salt from the Gatorade and then the manual shaking all work together to pull apart the layer of fat that keeps the DNA inside of your cheek cells. It's exactly like the concept behind using soap to wash dishes. It's just in a test tube and done a little bit more gently. But it's exactly the same like chemical process. Once the DNA is loose in the Gatorade mixture, it starts to dissolve and forms weak chemical bonds with the surrounding water. We have to add the alcohol to disrupt this bonding, which forces the DNA to undissolve and then become visible to the naked eye. Using cold alcohol is important because colder temperatures protect the structure of the DNA from falling apart once it's out of its protective shell. Once you have this extraction completed, you can really only hold it in the tube for like about maximum 5 minutes before it starts to dissolve again. But the cold alcohol will just shock it into being structurally strong enough to be like picked up and manipulated for a little while at least. Once you have this extracted DNA in a research setting or in a healthcare setting or whatever, we can do like a lot of different things with it. And this is what I do at my job currently, is a genetic analysis of traits. And so that first picture is a picture of a gel that has had gel electrophoresis performed on it. All of those bands are bands of DNA, essentially, and you maybe have done this in a science class before, but you just load one end of the DNA into a gel made of agarose, which is a seaweed-based sugar. Jello mixture essentially, and then run electricity through it, and the DNA will migrate according to size to the other side. You can also use extracted DNA to do forensic science, like identifying victims at a crime scene or perpetrators or paternity tests, things like that. We can do predictive testing. So like, for example, there's a gene called BRCA1, which is implicated in a lot of breast cancer cases. And so you can preemptively test your DNA for having that gene to know if you might be at risk for breast cancer later in your life. We can also use it to make clones and to genetically engineer organisms. And so that second picture is Dolly the sheep. That is the first successful viable clone that human beings have ever produced. She was cloned and she was born as a baby, and she grew up to be exactly genetically identical to her, to the organism that the sample was originally taken from. And then we can also use extracted DNA to generate medicine by using it to basically analyze antibodies that are produced by organisms during a natural immune response. And then we can pull out the genes that make those antibodies, put them into bacteria, and then use that bacteria to basically farm whatever like chemical we're trying to create. So like that's how insulin is created. A variety of other medications are created that way. And it's very cool science, although it's unfortunately not as doable in a classroom because you do have to be very precise and the equipment is quite expensive. But it is a cool application. If I were doing this in my classroom, I would probably go for the forensic science application route just because it's like fun to do like crime scene analysis. Like I'd probably fake like a blood sample or something and have them figure out who the perpetrator is based on reading a gel, because all of that can be produced fairly easily and cheaply without a lot of precision or expensive equipment. So yeah, these are my works cited. I essentially just went to this website about a suggested curriculum idea, which is the Seeing Your DNA science experiment for kids. And then on that statistic about how long our DNA is, it was this separate sciencefocus.com paper. So yeah, that's basically everything that I had. So I hope you guys are holding up okay. And I'm excited to see your presentations.